Next up is Peter, who is going to give us more specific information about the enhancements that have been made in adapting CyanoCyc for the cyanobacterial community. And there you go. Peter, take it away.
All right. Thank you, Lisa. Can you hear me?
Yes.
Okay, great. Yeah, and so I imagine you're going to send the survey link via email after the symposium is over, as well as a link to the video recordings of the symposium.
Yes, thanks. I forgot to
Great.
mention the video recordings. We will be making those available, and I'll be emailing everyone.
Great. So what I want to do in this presentation is to tell you kind of in more detail what CyanoCyc is and how it relates to BioCyc. And to do that, I need to introduce BioCyc as well. So BioCyc, as Lisa said, is a collection of 20,000 pathway genome databases. And each database has essentially the same structure, the same types of data that it can contain, although there's different databases have different proportions of how much of each data they actually do contain. But each pathway genome database describes pathways, reactions, metabolites, as well as the proteome and genome of the organism. And in some cases, they'll contain high throughput data sets like gene essentiality data or phenotype microarray data. And they can also contain gene ontology terms as well. These tier 1 databases have been very highly curated. For example, the E. coli database has been curated from 40,000 publications, and the MetaCyc database has been curated from 70,000 publications. So really rich collections of information. And the tier 2 databases are moderately curated. Typically under a person year of curation is where we draw the line between tier 1 and tier 2. And then the tier 3 are the computationally derived databases with no curation. So how do we create a BioCyc database? As one of the past slides indicated, it's really a combination of 3 types of information that go into a BioCyc database when we create a tier 3 database. We're performing a number of different computational inferences using software tools that our group has created to essentially extend the genome annotation. We combine the computational inferences with a number of data import operations. And then for only a selected number of databases, we perform manual curation to further improve the quality of the data. Now, in a bit more detail, what are these computational inferences that we perform? They're all present in the Pathway Tool software. They're all performed by the Pathway Tool software, and they will predict the metabolic and transport reactions present in the organism. They'll predict the metabolic pathways that those reactions give rise to. They'll predict what we call pathway hole fillers, or which genes in the genome might code for missing enzymes in these pathways. Because typically there are many pathways in any given microbe that are missing a number of enzymes. We predict operons, and just as an aside, we've actually developed a new version of our operon predictor that we just released that's improves on the accuracy of that task. We also have a predictor for protein complexes that creates new protein complexes in the database that are linked to their subunits. We also compute orthologs between the new database and all other proteins in BioCyc, and we compute PFAM domains across the organism, across the genome. The data import, we import data from a number of public databases. For example, from UniProt, we import both gene ontology terms and protein features. And it'll really depend what kind of data different databases have as to what's available to import. So those are the basic initial steps of creating a new PGDB. EcoCyc is probably the most extensive organism-specific PGDB. Another thing that we've developed in the past few years is the ability to propagate annotations in one of our PGDBs, a quote master strain, to other strains that we haven't been able to curate. For example, we started this a year or two ago for E. coli, is to propagate annotations from the highly curated K12 strain within EcoCyc to the 480 other E. coli genomes in BioCyc. And that propagation is done based on orthologs that we've computed between the genes in these different strains. And we're now starting to apply that procedure to other BioCyc genomes as well. I'm not actually sure if we've done it for Cyanobacteria, but we will get there. And then this is a rundown of the different types of information present in EcoCyc. We authored a paper called the Multidimensional Annotation of the E. coli Genome a number of years ago to try to emphasize that databases like EcoCyc and the other PGDBs annotate the genome at many different levels on many different dimensions, from protein complexes to protein features to metabolic pathways. And in EcoCyc in particular, there's really rich collection of regulatory information. And most BioCyc databases don't have very much regulatory information. EcoCyc is really ahead of the others in that way. Now, as someone mentioned, a really key database within BioCyc is MetaCyc. It is a really highly curated metabolic pathway database that contains experimentally determined pathways, reactions, enzymes, and metabolites from all domains of life. And we use MetaCyc as the reference pathway database that's used for predicting metabolic reactions and pathways in other organisms as part of the process I just showed you a few minutes ago. When we create new BioCyc databases, we're essentially recognizing pathways from MetaCyc that are present in the new BioCyc database. So as you can imagine, the more pathways we have in MetaCyc, the more complete a pathway prediction job we're going to be able to do. And I want to both present some more detail on how much data we have in MetaCyc and BioCyc, and also do a quick comparison to the KEGG database that many of you are familiar with. In BioCyc overall, we have 20,000 pathway genome databases for 20,000 sequenced organisms. In KEGG, it's 8,600. We have 69 Tier 1 and Tier 2 databases in BioCyc. In KEGG, we don't really know how to compute that, how to figure that out, and we're not even sure if KEGG has a notion of Tier 1 or Tier 2. And we can search both BioCyc and KEGG to find out how many publications are cited in the database, which we assume translates into information has been curated from those publications. And in BioCyc, it's 138,000 publications, and in KEGG, it's 75,000 publications. Now here I'm comparing the single MetaCyc database to the KEGG reference database. Both systems have the notion of a reference pathway database that's used to predict pathways in other organisms. In MetaCyc, we have 3,085 pathways. In KEGG, they have 425 modules, about 1/7 the amount. We have 18,000 metabolic reactions in KEGG, in MetaCyc, compared to 11,000 in KEGG. The number of metabolites is quite comparable. KEGG's a bit ahead there. And another real source of value in MetaCyc and BioCyc in general is what we call the mini reviews that the curators have authored about genes and enzymes and pathways and operons. we have 10,000 textbook page equivalents of mini reviews in MetaCyc alone. KEGG has 1,500 textbook page equivalents. Let's survey a bit more about the BioCyc data. I'm going to skip a few of these slides because of time, but we have a notion in BioCyc and MetaCyc of pathway variants, and these are alternative pathways that accomplish similar biochemical functions, or maybe even identical biochemical functions, but using different biochemical routes. So for example, we have 3 different variants of alanine biosynthesis. All 3 are actually found in E. coli, but one of them is not found in humans or B. subtilis. So the organismal distribution of these pathways does vary. And we have 7 different variants of the TCA cycle, all of which have similar but not identical sets of reactions within them. We also have a notion of super pathways in BioCyc, which are groups of pathways linked together by common substrates. For example, this overall pathway is called the super pathway. Well, I'm sorry, I'm not sure the exact name, but it is a super pathway that contains biosynthesis of charismate from deerythrose 4-phosphate, and then biosynthesis from charismate of phenylalanine, tyrosine, and tryptophan. So this one pathway can be shown on your screen, and you can also navigate to its subpathways. And you can define multiple levels of super pathways in BioCyc. All right, I'm going to skip a few of these things. I also want to give a quick summary of the many different informatics tools that are present in BioCyc. I'm not going to read through every bullet on this list, but we divide the tools into genome informatics tools, pathway informatics, regulatory informatics, microbiome informatics, comparative analysis. There are tools for transcriptomics and metabolomics data analysis, and there are a number of advanced data manipulation tools. And just to take you through one of these categories in detail, and one reason I want to mention this is because in an hour-long tutorial that Lisa's about to give you, obviously we can't cover all of these tools. So we do have recorded webinars on some of these tools, and we will be giving other tutorials in the future. And we have online help pages as well. So the pathway informatics tools include search and information pages for pathways, metabolites, and reactions, the ability to infer the metabolic network of an organism from its genome. We have zoomable whole organism metabolic network diagrams for each of the 20,000 organisms in BioCyc. There's a separate zoomable metabolic network diagram. We have a tool called the Metabolic Network Explorer that lets you interactively explore a metabolic network one step at a time in a forward or backward direction from a starting metabolite. There's a metabolic root search tool that should be particularly of interest to those of you doing metabolic engineering. It will compute an optimal route from a starting metabolite to an ending metabolite, either using only the reactions within one organism or adding select reactions from MetaCyc. We also have a tool for pathway-based inference of the nutrient requirements of an organism. And we have a set of metabolic modeling tools using flux balance analysis where you can convert a pathway genome database to a metabolic model. Okay, let me now move ahead, excuse me, and talk about CyanoCyc. So I've given you this overview of BioCyc as this collection of 20,000 databases. And what we've done, CyanoCyc is essentially a way of customizing BioCyc for the cyanobacterial community. And we've had this great series of interactions since last spring with our Cyanobacterial Advisory Committee, who are listed here. We've been meeting more or less every 2 weeks with them, and they've offered just a great series of suggestions. I want to give a big thank you to them. And in particular, Jeff Elhai has come up with just a huge number of new ideas for us to implement, and he's reported a lot of bugs, which have really helped with the reliability of BioCyc. We're always anxious to get bug reports if you run into problems. And yeah, Jeff's just offered a huge number of suggestions. So thank you, Jeff. And we look forward to continued interactions with all of you. Now, so CyanoCyc, one way to look at CyanoCyc is that it's a subset of BioCyc. It's the cyanobacterial subset consisting of 276 genomes, 4 curated databases. So we undertook curation efforts for each of these 4 organisms, all of which were literature-based. And we ended up curating almost 1,000 publications across these 4 organisms. And actually, more than 500 of the publications were in Synechococcus elongatus, so quite a few publications for that organism. Another way that we customized CyanoCyc is by creating the cyanocyc.org webpage. So it's a new entry point to BioCyc for the cyanobacterial community. And I'll note that a single subscription covers both BioCyc and CyanoCyc. You know, just as airplanes sometimes have multiple doors that you enter the airplane through, we have multiple entry points to BioCyc, but ultimately you're interacting with the same computers, the same basic website, the same databases. Some other things we've done to customize CyanoCyc, we've added 7 new genomes that the advisory board suggested. We've also added many new organism synonyms to our organism selector, because when you're working with BioCyc, one of the first things you want to do is select which organism are you going to search, which organism are you working with currently. It's called your current organism. And we were kind of shocked to learn that NCBI had a few years ago deleted many of the familiar names for cyanobacteria in favor of newer names in the NCBI taxonomy database. And that's the main database that drives the organism selector. I'll just show you really quickly what I mean by the organism selector. At the top of most BioCyc pages, you'll see this button, Change Organism Database. That's how you select which database you're going to work on. And Lisa will demonstrate that in more detail. But yeah, so we've restored, again, Jeff Elhai provided all of these synonyms. We've added back in synonyms that are going to be more familiar to people who have worked in the cyanobacterial community for a long time. We've also ensured that we have orthologs computed across all 276 of the cyanobacterial genomes, because we're perennially behind in computing orthologs for BioCyc. There are a number of organisms, tier 3s, that don't have orthologs computed, just because it is such a time-intensive process. And let me just show you really quickly one example of how orthologs are used. If you're on, say, a BioCyc gene page, in this case for the curated Synechococcus elongatus database is where I am, you can invoke the multi-genome browser that was shown in one of the talks earlier. And the one way to do that is if I go over here to the right and say change organism databases for future comparison operations, if I type in Synechococcus elongatus, so there are 4 other elongatus genomes in BioCyc. I'm going to select them all and click OK. And then I'm going to say align in multi-genome browser. And the software will compute the kind of view that we saw earlier on. Just wanted to show you how this is done. because the way this view is computed is by starting from the gene page, the gene for the gene page I was just on. The software is going to compute the orthologs for that gene across all other BioCyc organisms. Sorry, against the 4 organisms that I selected, and it's going to align the genomes at those orthologs. It's taking a little longer than it should here. I'll give another few seconds, and then I'm going to go on if it doesn't give me a result. All right, let's come back to that in a minute. We also made some extensions to the sequence pattern match tool within BioCyc. So PatMatch is a program for searching for short sequence patterns in nucleotide or amino acid sequences. And in the past, really the only way to run it was to search one entire genome. So you could enter a pattern, a series of nucleotides or amino acids, and selected wildcards, and run it against one genome. Well, some suggested extensions that we ended up implementing were the ability to define a specific set of protein sequences to search via what's called a smart table in BioCyc, which is a table of data that you can store in your account. We also added the ability to use PatMatch searches against a list of organisms, this should say organism proteomes, via a smart table. So the idea is that you can define a smart table that contains a list of BioCyc organisms, could be 3 organisms or 50 organisms. I think there's a limit of 70. And PatMatch will search your pattern against all those proteomes. So it's a way to do a much broader search than was possible before. Oh, okay. Another thing we've done is to create in BioCyc smart tables that list cyanobacteria with specific phenotypic properties. And again, these were all provided by Jeff Alhai. Thank you again, Jeff. And for example, there's a list of the unicellular cyanobacteria, picocyanobacteria, nitrogen-fixing cyanobacteria, etc. And we will add links to these smart tables on the CyanoCyc homepage or the Introduction to CyanoCyc page shortly. But really the way to get at those is, let's see, if you go under Tools and then Public Smart Tables, if you simply type Cyanobacteria list under the filter and click Filter, then it will show you only the public smart tables for the cyanobacteria. And soon, it's not very easy to do this now, but our goal is to make it so that you can use these lists in comparative operations within BioCyc, but that's not quite possible yet, but we hope to have that soon. Another thing we've done, also suggested by the advisors, was changing what I'll call the prominence of different accession numbers. You may be aware that over time, RefSeq actually has created 2 sets of gene accession numbers for many of its genomes. And what happened is that people got familiar with the initial set, and then RefSeq changed it and introduced this new set. A lot of the genomes do have both sets in RefSeq, but we had previously chosen the newer accession numbers to show in the genome browser, for example. And what the advisors told us is that, well, people aren't very familiar with those new accession numbers. Can we make the older accession numbers the default? And we said, sure. And so we've implemented that. And now when we look at the genome for a cyanobacterium, we will see the older RefSeq accession numbers. Okay, Lisa is taking you through this about information about our subscriptions. And I just want to note that really encourage you to approach your library at your university to purchase a institutional subscription, because it will save you money. It won't cost you a thing. The library will pay for this. And basically, the more students and postdocs and faculty members that a library hears from, the more likely they are to purchase a subscription. I also want to quickly mention our Pathway Tools software. So Pathway Tools is the software that powers the BioCyc website, and you can download it locally, and you can use it to perform metabolic reconstructions and perform the other computational inferences that I talked about describing how BioCyc is created. So you can feed in your own sequenced genomes. You can also feed in a set of mags to create pathway genome databases for a whole series of organisms within a microbial community. And the software is free. It's been licensed by more than 12,000 groups now, and it runs on Mac, PC, and Linux. And I think Lisa might have taken you through this as well, some of the availability, so I will skip this. So in summary, I've described the BioCyc Pathway Genome Databases. It's a collection of actually now 20,000 databases, of which MetaCyc is a special database because it's not organism specific, but is a non-redundant database of literature-derived pathways from all domains of life. And it's what is used to construct new BioCyc databases. And I gave you an overview of the tools within BioCyc, which fall into the different categories listed here. And Lisa's going to give you an introduction to those shortly. And so feel free to put any, okay, and here's some ways to learn more through the online BioCyc help and the webinars and a recent paper on BioCyc. And there's a paper on Pathway Tools as well. So I will stop there. Happy to take questions in the chat, but I want to maximize Lisa's time for the tutorial here.